Just make sure that, yep, it's coming through. 1, 2, 1, 2. And your audio, you're good to go. Perfect.
All right, good morning, or I guess we call it now afternoon, everyone. Today, I'm very pleased to invite Dr. Ahmed Abou-Setta to do the presentation today on the importance of systemic reviews prior to the conduct of clinical trials. Dr. Abou-Setta is the director of the Knowledge Synthesis Platform in the Center for Healthcare Innovation. And he has been there for how many years now?
Almost a decade now.
Almost a decade now. He is a clinician and a clinician scientist with extensive experience leading knowledge synthesis projects, including the preparation of systemic reviews, clinical practice guidelines, and comparative effectiveness reviews for a wide range of governmental and non-governmental and scientific institutions. Dr. Abou-Setta's areas of expertise includes clinical epidemiology, epidemiological methods for evaluation of quality of data, clinical trials, and observational studies, the meta-analysis of trial data, and methods for utilizing direct, indirect, and mixed evidence. Dr. Abou-Setta combines unique insights as both a clinician and a postdoctoral level researcher with research experience in various clinical settings. These clinical topics are broad and have included reproductive endocrinology, pain management in elderly patients with hip fracture, management strategies in hypercholesterolemia patients, and antipsychotic medications of adults with schizophrenia and bipolar disorder, management of male impotence and impotence and prevention of premature. Sorry, I don't have my glasses on, so I'm doing pretty good here, actually. Of ovarian failure in women undergoing therapy. So, on that note, I will let Dr. Abou-Setta take over and do his presentation.
Thank you so much.
Yeah.
Thank you so much. Thank you all for coming today. So, first of all, before I begin, thank you again for coming during your lunch hour and attending this simple talk. To talk about systematic reviews and their importance in clinical trials, especially in the preparation of clinical trials, you could write books on this. I mean, there is no end to the amount of information and the importance of conducting systematic reviews before conducting a clinical trial. Just a show of hands, how many of you have been involved in a clinical trial? Expected a lot. So how many of you have been involved in a systematic review prior to conducting a clinical trial? Okay, very few. And that's unfortunately more common. And we'll talk about why this is so important. So we begin with the whole systematic reviews in the area of knowledge synthesis that we live in today, and the idea of evidence-based medicine, from a statement that was made by Archie Cochrane in 1979, where he said, "It's surely a great criticism of our profession that we have not organized a critical summary by specialty and subspecialty, adapted periodically of all relevant randomized control trials." So even back then, in the 70s, they realized that there is a difference in quality and potential biases that can be introduced based on different study designs. So they acknowledged that randomized controlled trials were still the most important, but they also realized that we had not yet sort of categorized them, cataloged them, understood what's good about some, what's bad about others. And so generally, in your basic epi courses, you've seen the evidence pyramid, and generally we believe that if you're now in the realm of evidence-based medicine, that we've moved away from eminence-based medicine, or the oldest white guy in the room doesn't make the decision anymore, but rather we should be following evidence, where systematic review and randomized controlled trials are really on the top, and more of the databases and the textbooks and the expert opinion are on the bottom of the evidence. But even that's being challenged today, and the idea of a systematic review has been proposed to not particularly provide a hierarchy for what evidence is, but rather to put a spotlight on the evidence. So in other words, we have evidence from randomized controlled trials, we have evidence from previous cohort studies, case-controlled studies, and so on. What makes one better than the other? What are the potential methodological flaws in the research that was done before us? What were the potential biases that can have affected the results of research that came before us? And so the one thing that we generally all do, we all, throughout our lives, throughout our education, we've done literature reviews or literature searches. And to understand a systematic review, you first have to understand the important differences between them. So a literature review is generally just an overview of research on a given topic that answers related questions. It could be on dinosaurs, it could be on the World War II, or it could be on a specific clinical research question that you're interested in researching. The key characteristics, it organizes the literature, makes sense of it all, it connects those dots. It evaluates the literature. You have high quality, you have low quality, and we'll get into that in what makes some literature high quality and others low quality. It identifies patterns and trends. You can say, well, I've looked at all the literature and I've identified 100 studies, and 90 of them tell me that drug really works great, so probably that drug works great. It also synthesizes that evidence, so it tries to make sense and some sort of factor into it, like some sort of math into it, and like, what does that all mean? But it's not an anti-bibliography. It's not just like a list of information. It's not a one-stop shop for everything, and it's not a book review. And that's sort of just starting the stage of we've all done these in our lives, but we probably have not all done systematic reviews. So what is this, systematic review that we always hear about? So the definition, a coined definition of it is its attempt to identify all the empirical evidence that fits a pre-specified eligibility criteria in order to answer a very specific research question or clinical question through a reproducible and transparent process. And let me just stop here for 1 second. So we talk about you pre-specify, you have a very specific question, you're not going out to try to identify everything, it's reproducible and it's transparent. Now this is very close to what we do in a clinical trial. In a clinical trial, we ask a very specific question, we have to be reproducible, and we have to be transparent in how we did it. The main difference between the systematic review and the actual individual clinical trial itself is the systematic review is trying to identify all available evidence that answers that question, while rather in a clinical trial, we are using the patient that we have at our disposal or our catchment area to answer that clinical question. The key characteristics of a systematic review that, again, you have a clearly stated set of objectives with predefined eligibility criteria. It's explicit reproducible methodology. You have a systematic search that attempts to identify all studies that meet your eligibility criteria. You assess the validity of those findings. So you found studies that answer that question, but how valid, and we'll go into validity and why that's so important. But additional characteristics is we talked about earlier in the literature review, we could do some basic math in our head and talk about some frequencies, and the majority of the studies said this, or the characteristics of were that. But actually in systematic reviews, you can also couple it with highly sophisticated statistical methods that can both summarize the evidence itself, but maybe even more importantly for clinical trialists is it can tell you about the biases. And what do we know about the biases that could potentially have affected previous trials that have been conducted in research? And so just a quick overview again of how our traditional lit review that we've done throughout our lives, and you may be doing on a regular basis prior to conducting a clinical trial, how it markedly differs from the systematic review, where the systematic review is very focused, it's very transparent, you have pre-specified criteria because that prevents the introduction of bias, and we'll talk about bias in a little more. It allows for rigorous critical appraisal of what has been done before. The conclusions are based on a totality of the evidence, and you can talk about the grading of the evidence or the external validity, which we'll talk about again in just a little bit. So why should we care about these systematic reviews? First of all, and this is sometimes shocking to people, the primary research is often false or biased. And one of the most downloaded and read papers in all of medicine is a paper by Jadad, which talks about why most published research findings are actually false. And he goes into great detail about why his belief that most research doesn't truly come to par to answering the question that originally started trying to answer. So we know that primary research is often false. We also know that systematic reviews are highly publishable, even with negative findings. And often, you can't really get today a decent grant unless you prove to the funding agency that you've done your due diligence, in other words, conducting a proper systematic review to prove that you actually need funding for that clinical trial. And I've been on funding panels, and it often can come down to the point of not only just presenting the evidence, but actually saying, has this been peer reviewed? Have your peers actually said, yes, we have a gap in the literature. We need another trial to answer this question. And most funding grants for clinical trials will not be accepted with a systematic review showing the need for primary research. And this has led to a huge shift in the number of publications that we see of comparison of randomized controlled trials and systematic reviews, whereby if we look at the blue line, the trials or publications of randomized controlled trials has steadily grown over the years naturally in Medline, but systematic reviews have quickly overpassed it since 2014. So now we're publishing actually more systematic reviews of research than we are of randomized controlled trials. One of the important things we talked about is when you go to a guideline committee and you say, hey, I need funding for this new trial, and they say, great, where's the evidence that you actually need funding? Where is the evidence that we truly have a gap in the literature? So this was an analysis looking at mortality outcomes, and then they mapped it out against the year that the trials were done, number of participants. And it clearly shows you that by the 15th or 16th trial, we had a clear signal saying that there was benefit of the drug. Having said that, another 50 trials were conducted with thousands of patients randomized that were needlessly randomized because the answer was already evident many years ago. But that evidence was never presented. So we talk about this traditional systematic review and how it's important. And so, what goes into that systematic review in a nutshell? Very quickly, we start at a baseline. Whatever I know today is my baseline. I do a literature search, preferably using a clinical trial specialist or a librarian, somebody who actually knows how to search databases. You're going to select studies through citation screening and full text screening. Maybe most importantly, even for clinical trialists, is you're going to conduct quality assessment, risk of bias assessment. You're going to extract data. You're going to analyze that data, which can also potentially show you other biases. You're going to grade the strength of that evidence, and you're going to present that result. And what that does is it brings you back to a new baseline. So a systematic review is an ever-evolving sort of cycle of knowledge. As you produce more knowledge, you come to a new baseline. You have a new understanding of the question that you are simply going to answer, because that's what we do in clinical trials. We have a question we don't have an answer to, and we go out to try to identify that evidence. That evidence may be already there for you, or it may be there but extremely biased, or there could be other factors that affect it. And we're going to talk about that. So the two things we're going to really sort of hammer on today, due to time, is the quality of the assessment and the potential for biases that can be introduced or identified by systematic reviews that occur in clinical trials. I'm going to stop right here for a second and ask if there's any questions. Did anybody understand anything? Okay, so let's throw some more stuff at the wall and see if anything sticks. Okay, so systematic reviews to determine the validity and the potential bias in prior research. So that really comes key. And again, I could talk for hours about how systematic reviews can be involved in clinical trials, whether it helps us to identify patient populations, whether it helps us to identify where there are gaps in the literature that we don't have evidence. We can look at it in things about equity. We can talk about patient preferences. We can talk about lots of areas that systematic reviews can be used. But just for the sake of time, we'll talk about two important aspects regarding validity and potential bias of previous research. So when we talk about validity, we have two real sort of main goals of validity. We can look at the internal validity of a study that was previously done. And so we say, should we completely trust that study's findings? Great, somebody's done that clinical trial. Somebody's already done a randomized controlled trial on the same question that I want to ask. Should I still go forward and do that again? Or should I accept those findings results? So the internal validity are questions related to, should I completely trust the study's findings? Are there any certain factors present that may deviate results from the truth? While external validity is great, I may have a study and I may completely believe that that study works and I believe in the findings of that study. But is that generalizable? Is that information applicable to other patient populations? So how similar is that to study patients, interventions, comparators, settings, outcomes being measured in real life? Okay. So I'll give you a very quick example. There was a study that was done on pregnant baboons. And they showed that pregnant baboons, if they took this drug, it worked really great. As long as the pregnant baboon got this drug, the baboon never went into labor. Perfect. It's a great drug. Let's just spread it out and start using it in real-life clinical studies. And so therefore they tried it in a clinical trial. And it did not work at all. It was no better than placebo in real life. And so therefore I might say, oh, well, this study was done on a pregnant mammal, therefore it should work in pregnant women. It doesn't work that way. So therefore the study's findings might be completely valid in baboons, but is not generalizable at all in human subjects. So how much faith do I have that future research also will come to the same conclusions? Often pediatric researchers will often tell you that children are not small adults. So just because a trial was done on Tylenol in adults doesn't mean that it's going to work on Tylenol in children, and so on and so forth. Same thing with men and women, same thing with multiple different genetic disorders, same thing with geographical populations. So talking about the internal validity, whenever we look at studies, we look at three primary things in internal validity, and there are many more, but these are the real primary things. Is one, we look at the reporting, we look at the quality assessment of it, and we look at the risk of bias. So when we talk about reporting, really depends on what your trial's study design was. So was it a randomized controlled trial? Was it observational? Were you looking at case reports? Were you doing animal preclinical studies? What were you doing? There's a different tool for that to try to improve the reporting. So it's not how you did the study, it's not how that study was conducted, but just how it's reported to have been conducted. The more common thing that we are generally sort of in tune with is this idea of quality assessment. And we always think, well, high quality versus low quality, and then if it's high quality, I believe what they say, and if it's low quality, I question what they say. So what is this quality? So one of the definitions of quality was the extent that the study's design and the conduct can be shown to protect against systemic bias, non-systemic bias, and inferential bias or inferential errors. So again, we're going to study designs themselves, depending on the clinical trial that you are going to do. Not all trials are created equally, and I won't go into the biases and the potential qualities of cohorts and retrospective versus prospective, case-controlled versus randomized, and so on, because that's a whole that's a whole course on its own. But just the idea that you need to know that every study design itself carries potential quality issues or biases that can affect it, and the idea is how well was the conduct of it done to protect against these biases from entering into your study, both systemic and non-systemic. But quality is often confused with bias, and they're not the same thing. And now more and more we're moving away from assessing what is the quality of research that was done, but rather what is the potential for biases that can affect the results. But both bias and quality can make up internal validity. And one of the biggest problems with quality over the years has been that every group, every expert has sort of come out with their own idea of what is quality, what makes up quality, how do you say something is high quality versus lower quality. So Haddad scale, has anybody seen the Haddad scale before? Yeah. So Haddad was, in his time was actually a really great scale. He was a surgeon and a researcher, and he sent out emails or it wasn't emails time, it was snail mail. He sent out mail to all colleagues who did clinical trials, and he said, okay, tell me what would you consider to be the characteristics of high quality studies. And so they all sent him back, and he collated the information, and he came up with 3 main criteria to state, if you do this in your trial, then you are high quality. And it was randomization, so a randomized controlled trial, that you blinded, and that you reported on your follow-up. So let's walk through very quickly how this would be assessed. And let's assume we're looking at somebody did a randomized controlled trial, and I want to use the Haddad scale to determine whether this trial was actually high quality or not. And he very simply says, okay, was it reported or described, and I want to highlight that, was it reported or described as a randomized controlled trial? So in other words, they use the words like, we randomly assigned, we used random selection, we used randomization. If so, give it a point. If not, just give it a 0. But additionally, he knew that trials may claim to be randomized, but at the same time, they were quasi-randomized. And so let's just do a little experiment here and see if this is a true randomization or not. So let's go row by row, and let's say intervention, control, intervention, control, intervention. You're a mind reader. Okay? This is alternate randomization. This is not a proper form of randomization. If I told you whatever whoever's hospital number is an odd number gets the treatment, whoever is an even number gets control. Okay? Again, anyone can identify what group they're going to be in. So this is not a random form of randomization. So he still identified that and said, okay, you know what? If the trial reported itself like that, then no, we're not going to consider it randomized anymore. This isn't a true RCT. So let's take away that 1 point that we gave it because it said it was random. If they properly randomized, like they said, I use a computer-generated randomization or a random number table and so on, let's give it another point. And if they didn't describe it well, what can we do? Just give it a 0. So we're not going to duck it any points. We're not going to give it any more points. And he keeps going through the same thing, like let's do double-blinded, because they believe that double-blinded, placebo-controlled trials were the greatest thing since sliced bread. So did they describe themselves as being double-blinded? Yes. Then they give it a point. When you look at their double-blinding, did you really think that would have held true? So an example, for example, if I randomized people to a blue pill and a red pill, they probably know what they were getting, as long as they knew what red and blue were. But at least they could identify what study. Same thing if somebody was getting an injection versus a tablet. They probably knew what they were getting, even though we supposedly randomized them. And there are ways around that, using double dummies and sham interventions. But he says, well, if you adequately described it, I want to give you another point. And then if you didn't describe it, I'll give you a 0. And, if it was inadequate, so they said they used a dummy, but, obviously it wasn't proper, so we'll take away that point that we gave them for double-blinded. And last but not least, did they describe their withdrawals? The one key thing about all this is that they talk about did they describe, not did they actually do. So if a person is very conservative in their description of what they did, they can get high points. If they're upcoming or forthcoming and tell you everything they did, you might actually start taking away points and give them a lower quality scale. So, for example, if I claimed I did a randomized, double-blind, placebo-controlled trial in my title, I just got 3 points. And the average score for a randomized controlled trial is 3 points. So we knew that there are so many scales and they're not created equally. And formally, in 1999, Juni published a paper in the Journal of the American Medical Association where he compared 25 different quality scales on the same exact 17 clinical trials. And not surprisingly, he looked at 3 key domains: allocation, concealment, blinding, and handling of withdrawals. And Not surprisingly, they were all over the board. So in 28% of your scales, if you had a high quality according to the scale, therefore you had an effect on the outcome. In another quarter, if you were a high quality, there was no difference, but if you were low quality, then you had an effect. And in half the scales, study quality didn't affect. So the take-home message is, with regarding to study quality, it really just, it's a crapshoot. It's really, what trial do you have, which scale are you using, and what does it really mean? We really don't know. So we've generally moved away from looking at quality. Now we primarily look at bias. And what is this bias? Well, naturally speaking, as human beings, we are biased. Okay? We all have preferences. We all have beliefs. You wouldn't be doing a clinical trial if you thought it doesn't matter. You wouldn't be here today if you thought it didn't matter. I hope you're all here because you think it matters. But so what is this bias or this risk of bias? So in 2011, I did a study looking at how many trials in the Cochrane library, so including Cochrane reviews, did the systematic reviewers actually assess to be at low risk of bias. And you'll notice that primarily it's flatline. Like, we have not really had any market improvement in clinical trials. We did mostly randomized controlled trials. Until the 2000s, we barely start edging up. But what it does notice is the green line, which represents your unclear risk of bias, and your red and your little sort of like pink line here, which shows your high risk of bias. As reporting has improved, we've realized actually when people don't report things, it usually means they're at high risk of bias. So when people don't report that they used a placebo, it's probably because they didn't use a placebo. When they didn't say how they randomized, it's probably because they didn't randomize properly. So therefore, it makes us think about those trials that others have done before us that we're looking and saying, oh, well, that question's already been answered. We don't really need to tackle it anymore because so-and-so already did this really great trial. And then we start looking at, well, how did they do their trial? And we realize there's a lot of questions that are unanswered. And if those questions had been answered, often they will tell us that they are a higher risk of bias than we originally thought. So what is this bias? Before we actually go into what bias is, let's say what it's not, because there's an awful lot of confusion about it. So bias is not imprecision. So you're not biased if you have a small sample size. Sample size is related to precision. How accurately will your point estimate reflect the truth? Quality. So we already talked about quality before. Bias can occur in really well-conducted trials, and not all methodological flaws will introduce bias, and not all biases will decrease trial quality. And the easiest way to think of it is pharmaceutically sponsored trials are often of very high quality. They're probably the best quality studies you're ever going to find. And there could potentially be a huge bias in all of them. Not saying there is or there isn't, just saying there's a potential. Reporting. Whether I report, underreporting or poor report doesn't equate to quality or bias, because bias is related to how the conduct was done. So if I didn't tell you how I did, you're only really guessing at what I actually did. So if you say, I said I did a randomized controlled trial and I didn't mention any further how I randomly assigned participants to either of the groups, it's only a guess. I could have properly done it. I may have not done it. So it doesn't affect your bias per se. So a definition of bias by Cochrane is it's a systematic error or deviation from the truth in results or inferences. And the bias can go in either direction. Different biases can lead to underestimation or overestimation of true intervention effect. And the simplest way to think of it is I could have a bias that makes my drug look more efficacious than it actually is, or I could make it look safer than it actually is, or maybe not as harmful as it looks in real life. Oh, sorry, this slides over again. So we know that there's bias, and we know the results themselves of trials can be biased. So what's going to affect these biases? So we barely touched a little bit about some of the external factors. We said the source of the evidence itself is biased. So we know that pharmaceutical-sponsored trials lead to more favorable results, and conclusion, which is often mentioned as industry bias. And Cochrane has a strict policy of thou shalt not touch industry money, so that therefore your evidence itself cannot potentially be considered to be biased. But also maybe more dangerously, because that's easy to see. That's easy to see. There's an expectation if you're getting funded, you have to clearly state what your funding sources are. But maybe even more dangerous and more insidious are the internal factors leading to bias. Academic success. Now, we often think of if you're doing well in your career, that's a good thing, and therefore we should trust you more. But that actually may also lead to its own biases. So, has anybody heard of the drawer effect or the desk drawer effect? Okay. And it goes something like this. I'm going to do a clinical trial, and I get the results, and I don't like what I'm seeing, so I open my drawer, I put the results in my drawer, and I close the drawer. It's a desk drawer effect. Okay. Also, if I have published 9 trials that have shown this drug to be successful, and my 10th trial shows that it's harmful, am I likely to publish or perish it? Okay. So we are generally under pressure as academics to publish in big journals, to get headlines and so on. So there's a lot of pressure, and that may lead to biases. Grants. You want a grant, you need to have a good track record of publications, you need to show success, and the best way to do that is to present positive results. Senior advancement, same thing. You want to advance, you want to become a professor, you have to prove that you're an expert in the field. You do that by publishing more, you publish more, by getting more money, you get that, and so on. And you see there's a vicious cycle that's growing. Authorship bias. An author bias or authorship bias is one of maybe the most insidious and difficult ones to sort of put your finger on, because it's almost impossible to, you can easily suspect it, but very almost impossible to actually prove it without an investigation, usually by the university or the hospital that's doing where that has the authority to actually go into files and look at how research was done. It's a non-scientific form of bias, where the investigator's prior knowledge, beliefs, opinions, academic pressure to publish, or relationships, whether it be financial or professional, systemically confounds the presentation of results and conclusions. Okay? It's a lot to take in, but think of all these biases that we've talked about previously, all munched in together, and that can affect the way we present our results. But also, even on even the most well-respected individuals that we know and we hear about can be affected by bias. And this is a little small example that I like to talk about. I call it the wall of shame. So let's start off with our first specimen on the wall of shame, William Hannon, MD, PhD. He was a pilot at United Airlines. He was the co-director of the College of Aviation Center of Excellence for Simulation Research at Western Michigan University. He had 9 publications in PubMed, including U.S. government reports and millions of dollars of grants. Looks like a face value, very respectable person. Only one problem. He was never a doctor. He actually faked a medical degree and somehow moved up the line to becoming a head of research and conducting research and getting millions of dollars of grants. Well, must be an offshoot. So let's talk about Joachim Bolt, professor of anesthesiology and internal care medicine, hundreds of publications, considered one of the renowned experts of the time on resuscitation protocols. Unfortunately, Bolt was also one of the leading individuals that has had retracted publications due to different forms of authorship bias, fabrication of data, conducting trials without ethical committee consent, conducting trials supposedly with a drug that wasn't actually available in the hospital at the time when he was actually there, and so on and so forth. And again, these are the experts that we're talking about here. And Scott Rubin, another professor of anesthesiology and pain medicine at Tufts University, numerous publications, and considered at the time a renowned expert in pain management, especially multimodal pain management, and unfortunately another fraud with a lot of data fabrication, a lot of data manipulation, and really one of the people that set back multimodal pain management for years. And these were considered the icons. These were the people that they did clinical trials, and we looked up to them and said, wow, these guys have done the work. We believe in what they've done. And so one of the things that we recently, it's coming out next month, a paper that we did is trying to actually say, can we, using a systematic review of meta-analysis, try to flag these? Again, it's almost impossible to prove these things unless you have the power of authority to get into the clinical trial files themselves and look at what has been done. But can authorship bias actually be detected in a meta-analysis or a systematic review? And so what we actually showed is using meta-regression, you can actually look at how closely the results of clinical trials are from specific groups. So a trialist may publish multiple studies, and this is one of the things that started flagging with people like Bolt, is their sort of cluster was so different than everybody else's. So they're saying, everybody else is saying harm, 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 and he's saying benefit, benefit, benefit. And so we showed that with actually some statistical techniques like meta-regression, and there's people who have used other techniques, including Bayesian analyses and bootstrapping, to actually try to sort of flag signals. One of the people, I apologize, I'm losing his name at the moment. He was an anesthesiologist, and he challenged another prominent anesthesiologist on his work, especially in antiemetics, and sort of the take-home message was, prove it. And that's what he did. He went and he learned the statistical techniques. He went through the process of going through all the research that individual had published and presented evidence to say that the chance of him actually randomizing these individuals was something like 1 in 5 billion. So either he got really lucky in all his participants over the years, or there was fraud, and that started an investigation, and then the investigation actually proved the fraud. So moving on to, or more like wrapping up. So when we've been talking about bias and how bias can be introduced, and how do we look at a paper and say, yeah, I trust that study. I believe that the results truly are correct. To understand that, we have to sort of walk through the idea of a clinical trial and where potential biases can be introduced along the line. So we start off with a target population, as in any clinical trial, and we want to include participants, and we're going to randomize them, and then those randomization will go into a treatment arm and a control arm, and we're going to follow those up, and then we're going to publish our results. Pretty standard, pretty straightforward, nothing out of the ordinary. So where do the biases sort of enter into all these? Well, starting with, how do I select my population? Even the selection of the population, again, we talked about is randomization proper or not, so therefore can the next participant know what they're going to get? So can they potentially be included or not? And so today, with sophisticated and the price of some of these management tools have decreased markedly. But still, till today, we often will randomize people by alternate randomization, availability of, for example, the surgeon or the ultrasonographer, because of ease of randomization. That in itself can introduce biases, because the people who are selecting participants to enter can say, hey, you know what, come on Thursday because that's when the ultrasonographer is going to be there, therefore you'll get ultrasonography, rather than, for example, getting no ultrasonography. Or they might say, as an example given to me many years ago during my postdoc by a person who's now head of one of the institutes at CIHR, when he was a resident, he found his more senior residents taking an envelope and putting it up to the light and trying to see what's inside of it. And he says, what are you guys doing? And he says, oh, no, we have a patient and he's part of this trial, and we want to make sure he gets the intervention. So they're looking at what the envelopes say to make sure that he either gets the intervention or doesn't enter the clinical trial. Fast forward, they actually realized the intervention actually caused more harms than good. So even though they had a goodwill in trying to get the patient into the trial and get him on the intervention rather than the control, they were actually harming that patient. So we randomized our patients, we properly randomized, no problem. But another sort of facet to the randomization is We can create a proper randomization scheme, though. So I'm going to sit here on my computer. I'm going to jot up a nice little randomization scheme. But there's 10 people who are recruiting patients. So how do I let everybody know? So I'm just going to print it out and put it on the clinic wall and just check off the next person. So you obviously know, even though the scheme is randomized, but you obviously know who the next patient is going to be. So that can bias whether they get the control or the intervention. Properly randomized, nothing wrong with there. We're all good. We move on to performance. So we often hear that double-blind, placebo-controlled trials are the greatest because if we don't know what we're getting, then we can't change our lives. We can't change the way we practice, what drugs we take, what we eat, and so on to affect it. But also the physicians themselves. So imagine you're having a randomized controlled trial, and they can offer people are getting heparin versus a placebo, and a patient comes in with bleeding, and they know that patient's on heparin. They may be more likely to start going for another intervention or giving blood or platelets because they know they're on heparin, rather than saying, you know what, they're on a placebo. Let's just wait it out and see what happens to the patient. Let's watch the vitals. So therefore, even the clinicians themselves who are treating the patients in that clinical trial may make judgment calls based on knowing what that intervention or comparator was that the patient was getting. Detection bias is we know from lots of research that often an independent group looking at the same evidence may come up to a different conclusion than the PIs themselves. And often you'll see this in large clinical trials where they'll have a separate committee that evaluates safety, even efficacy, especially in oncology trials. And often the results will be similar but not identical to what the individual trialists have picked up. Also, in many clinical trials, especially multi-center clinical trials, they'll take samples and they'll send it to a central laboratory where everything gets analyzed centrally to make sure that you have the same baseline of comparison against all. So even detection bias can be introduced. Attrition bias, so Jadad picked up on this and he said, did they describe the dropouts? So dropouts are extremely important, and how people describe how they analyze the data. Has anyone heard of an intention to treat analysis or a per protocol analysis? So really quickly, an intention to treat is if you were randomized, you get analyzed, and if intention to treat is if you were randomized and you maintained the study and you met a certain amount of, you didn't have any protocol violations and you had a certain amount of compliance, then you would be still analyzed. The problem with that, and here's where the huge bias comes in, is what if somebody takes a drug or an intervention and they were uncompliant? And I'll give you a personal story. I was once, my physician asked me to take a drug, and the known side effect of that drug was metallic taste. Has anybody had something before with a metallic taste? Yeah. I was very uncompliant. I'll be right there. I took, I think, the first day, and that was it. And because metallic taste makes your life a living hell, you are now permanently sucking on a rusty pipe for the rest of the time that you're on that drug. When you drink water, you're literally drinking a pipe. You're eating, you're drinking a pipe. So it was horrible. My quality of life dropped, and I was like, I'm not on that any longer. My wife was stronger. She kept going. Kudos to her. I'm weak. If I'm part of a randomized trial and they say, oh, you're not compliant, we're not going to look at you if the infection went away or not, therefore, it could bias the results, saying, oh, yeah, the drug really works great in everybody who stayed on the trial. That percentage might be very low. There may be only 25% of the people who are compliant and get analyzed in the end. So it can give you a very biased view of the evidence. Last but not least, reporting. We talked about how I can just not report my results at all, or I report my results in a way that makes it look better than it seems. So I might say, like, in my unbiased approach, when I go to my grant committee and I say, I want to do a trial and I want to say whether, this drug actually really saves lives. That's what I really want to do. But then when I actually look at my results, I realize, oh, well, you know what? We're not actually saving lives with this drug, but there's some good signs. The blood pressure was better controlled, but we really didn't save lives. So all of a sudden, you switch your intention of your trial, and it becomes a trial about maintaining blood pressure rather than about saving lives. Okay? And that changes the purpose of it. So can I believe that if my intention is to save lives, can I trust these results? So there's a lot of play and introduction, or I don't report on it. Quality of life wasn't really, measured properly, and I'm going to make all the excuses in the world why I'm not going to report on it. And my clinical coordinators really didn't understand how to ask the question. So you know what? Yeah, those are just biased results. I'm not going to present it. When actually the quality of life may be a lot worse in the intervention that I want to make sure looks good in the end. Okay, last but not least, any questions? Okay, I really don't have much more to say. So, like I said, just a quick cap. This is only one aspect of why systematic reviews are so important for clinical trials and important to be done before a clinical trial is done. It tells us about what has been done before. Can I really trust what's been done before? Often the answer is no. Often the answer is what's been done, we can still improve on it. But also, is the question already answered, and we trust what's already out there? And really, what we're doing is really redundant and doesn't answer anything new. Thank you so much. Yes.
One of the areas of bias that I've seen is in scales.
Yes.
Clinical trials. There's one called the mental state exam or the full scheme that we use in psychiatry a lot. And there's a lot of times where it's statistically significant, but it is not clinically relevant.
Yeah.
The patient's still normal, regardless of the 3 or 4 point increase. Maybe you can perhaps comment on that, where it might be statistically significant, but not clinically relevant.
Yeah. So one of the things about working generally in methodology is it teaches you how to be a supervillain. We learn so much about studies themselves that we can set up studies that will provide us with the evidence we want to see in the end without actually doing the study. And that's by understanding what previous studies have done. So as you mentioned, I can take a scale that might not have clinical relevance but looks good on paper because I want this drug to come out looking good. And so you can bias your populations, you can bias doses of interventions, you can bias doses of control, and there are many examples of this, or how I'm measuring the outcome. So maybe a traditional way of measuring in psychiatry is I'm going to measure over several different scales and validated scales, but maybe I go off and I choose one that's not a validated one, or one that I made last weekend because I was free. But I can make it in a way where I know that the results will come out favorably to what I want. Yeah.
Very much.
Thank you.
My question, and then you are interested.
Yes.
There are out there. And the result has
Yes.
collection of these reviews in this direction where it goes in the opposite direction, and you want to make your judgment on what is available before you start the clinical trial. Please, how do you going forward in this situation?
Yep. So a systematic review, even again, on the pillars of it, it is supposed to be collecting all evidence. It is supposed to be transparent. It is supposed to be reproducible. And it's supposed to be there to limit the biases that can be introduced. Having said that, when we look at systematic reviews themselves, we have to assess bias. We have to assess quality. We have to assess reporting. Because unfortunately, because of the popularity of systematic reviews, everybody out there is now doing a systematic review or claiming to do a systematic review. And as an editor and as a peer reviewer, I get a lot of quote unquote systematic reviews, which I question whether that actually falls under the category of systematic review or not. Or I present a lot of questions on transparency. How did you do this? How did you do that? What about the presentation here? What about that? And the other reviewer could be just completely saying, oh, this is a really great study. We should publish it tomorrow. And I think to myself, were we actually reviewing the same paper? And I talk to people who are methodologists, and they get this very common. So just because you see something in print, again, don't assume, therefore it's high quality, including based on the journal. So don't say, this was published in BMJ or CMAJ, therefore I present the truth, and this one is presented, in a North Korean journal, and therefore it's biased. Thank you. Thank you so much. How was it? Thank you for sending me the email.
Just wanted to make sure, because that's not PSC check.
Yeah, I was like, I've read that before, but I don't remember where. I was like, yeah.
I want to make sure.
You want a copy of it for? Give me email too. Oh, yeah. Well, no. Okay, thank you so much. Thanks for inviting me. Thank you. Yeah. That's a great way to run. But still, there are certain things which